Welcome to a new episode of Drug Interactions. Today, we're diving into answering a crucial question. What happens if you react too early and too aggressively to a drug interaction before the time course of the significant interaction takes place? Let's dive into a case involving a 29-year-old patient who presented with epileptic seizures. She had severe iron deficiency anemia and was started on levetiracetam, which was discontinued shortly after due to elevated liver enzymes. The patient was then discharged without anti-epileptic medication, only iron supplementation. A few days later, the patient returned with generalized tonic-clonic seizures, progressing into status epilepticus. Lorazepam and phenytoin were used to manage her seizures. Less than 2 days after phenytoin initiation, the patient was started on lamotrigine. The dose was 50 milligrams twice daily, which is 4 times the usual initial dose of lamotrigine without enzyme inducers or inhibitors. Around 13 days after starting lamotrigine, the patient developed severe, profound neutropenia, with an absolute neutrophil count dropping to 800 and then to 0. Investigations ruled out autoimmune and infectious causes, and it was determined that lamotrigine was responsible for this neutropenia. Lamotrigine is known to cause hematologic toxicities, including leukopenia, thrombocytopenia, and neutropenia, as part of its hypersensitivity spectrum. But why did this happen? The answer lies in the drug interaction and improper timing of the lamotrigine dose escalation. Lamotrigine was escalated too early and too aggressively, especially in the context of a potent enzyme inducer like phenytoin. Here's the key point. Phenytoin, an enzyme-inducing anti-epileptic drug, requires time to reach steady state, which typically takes 5 to 6 days. However, enzyme induction doesn't just stop at steady state. The induction process itself, meaning the increase in metabolic enzyme activity, takes additional time, usually 1 to 2 weeks after steady state is reached. This is because the induction of genetic transcription of the enzymes needs time to upregulate them to a new steady state, which then requires substrate dose escalation. In this case, lamotrigine was started at a higher than recommended dose before phenytoin's enzyme induction had fully developed. As a result, the patient was exposed to much higher levels of lamotrigine than intended, leading to hematologic toxicity, particularly neutropenia. The lesson here is twofold. First, always account for the steady state of enzyme inducers like phenytoin, and second, consider the full time course of enzyme induction, which requires time after steady state for the enzymes to be fully upregulated. Escalating doses too early, before the induction is complete, can lead to dangerous overexposure to drugs like lamotrigine. Eventually, lamotrigine was discontinued, and the patient's blood counts recovered after supportive care. This case underscores the importance of respecting the time course of drug interactions, particularly when dealing with enzyme inducers. In summary, always be mindful of the time needed for enzyme induction when adjusting doses, and avoid reacting too early to potential interactions. Rushing this process can lead to severe, preventable complications. Thanks for tuning in, and remember, understanding the timing of drug interactions can prevent life-threatening outcomes.